Our third speaker for this session is now going to focus on herbicide resistance, and it's a great pleasure to introduce my colleague from the University of Adelaide, Professor Chris Preston. Chris has been working on understanding the management of herbicide resistance for the larger part of his career, and his current projects include looking at integrated weed management in southern farming systems to combat herbicide resistance, improved herbicide efficacy and efficiency and longevity in southern no-till farming systems. He looks at mechanisms, evolutions and inheritance of resistance, and he also chairs the Australian Glyphosate Sustainability Working Group. So over to you, Chris. Thank you.
Thank you, and it's it's really nice to be able to get out of South Australia after the last couple of years. And what I want to run through this morning is primarily talking about some recent survey results of herbicide resistance, and then I want to touch on some recent findings about resistance to glyphosate and resistance to the phenoxy herbicides, and perhaps how that might think our management going forward. So to start off with, I'm going to talk about a resistant surveillance project that the GRDC has funded across Australia, and in the summer of 2021, we surveyed, and when I say we, it was a lot of people, we surveyed fields across Australia from Geraldton up into central New South Wales for both winter weeds over all of that, and for summer weeds in northern New South Wales and Queensland. Weeds were collected out of these fields, and we've been testing them for resistance to herbicides. And not all the data's in yet, but I'm going to show you a snapshot of some of the data that we have. On this map here, what I've got is all of the fields that were visited have been located on that map. The ones that are in blue are where we found annual ryegrass. And so you can see that annual ryegrass is present a lot across a lot of that area. But once you get into northern New South Wales and into Queensland, it's a bit sparser, but some of that's because some of those were also summer weed surveys as well as winter weed surveys. So starting with ryegrass, what I've got here is a summary of the data. The ryegrass data is now complete, so I've got a summary of the data that we've collected on population to resistance, and the scoring system that I'm using here is that when we test these populations, 20% of the plants have to survive the herbicide before we call it a resistant population. That's really to do with what a farmer might notice in the field. So, there's actually probably more resistance than I'm showing here. So what we've got here is I've got the post-emergent herbicides. So these are the herbicides that we put out after the weeds and crops have come up typically, and these used to be the mainstay of ryegrass control in Australia. Certainly they were when I started my career. So running down the herbicides, Axial, the Group 1 herbicide, what we find is it doesn't really matter where we go across Australia, we've got high levels of resistance to that herbicide. And fundamentally, we can consider that segment of the Group 1 herbicides really just don't work on ryegrass anymore. There are still some populations out there where it operates, where it'll work, but most of what we're looking at, it doesn't work. Clethodim's also a Group 1 herbicide, but behaves a bit differently. What we'll often find is that we've got resistance to things like Axial, we can still use clethodim. What we're starting to see now is we're starting to see relatively higher rates of clethodim resistance turning up in these surveys. And I will note that we're actually spraying these plants at the ideal time, 2 to 3 leaf stage, under ideal conditions, with the full rate of clethodim. Often farmers in the field are trying to apply clethodim late, so they're applying it to large tilled ryegrass. And clethodim resistance has both a rate response and a plant size response. Moving across, we've got the Group 2 herbicides, and it's probably fair to say that the Group 2 sulfonylurea chemistry is just not ryegrass herbicides anymore. We've got really high levels of resistance to those herbicides, and a lot of that's flowing across to the imidazolinone herbicides, so things like Intervix. are not really controlling ryegrass. So they're products that we're going to be largely using for other weed species than ryegrass. Glyphosate, we're starting to see glyphosate resistance turn up at higher frequencies in our survey than we've seen before. It's becoming common. So it used to be rare, and certainly I spent a lot of time as chair of the Glyphosate Sustainability Working Group to keep it rare. We now no longer have a Glyphosate Sustainability Working Group because glyphosate resistance isn't rare anymore. But it's still, you know, there are areas where we're not finding any glyphosate resistance, but there are areas where we're finding quite a lot. In the survey here, paraquat resistance, we haven't found any in our survey results. That doesn't mean that it's not present. We know we actually have some paraquat resistance in ryegrass, but it's fairly localised at the moment, and that means that we just don't pick it up in this sort of survey. So the results of this survey, I think, confirm what we've known for quite a while about ryegrass, is that ryegrass management in terms of herbicides is now largely a pre-emergent herbicide space. That's what we do. So we're looking at pre-emergent herbicides as well. And in the pre-emergent herbicide space, at least as far as the survey goes, things are looking quite a bit better in terms of how much resistance we've got. Triflurin resistance is probably the worst case, and South Australia and Victoria lead that. We're getting a little bit of resistance to the Group 15 herbicides, particularly the Boxer Gold collection, so Avidex and Boxer Gold and those sort of products. That's not necessarily flowing over to all the other Group 15 herbicides like Sakura. Okay, so Sakura's still working in this survey. Propizamide, we're using roughly here for this particular test, but propizamide is still working on ryegrass, and it's one of those really interesting stories because it's in the same group as triflurin. Triflurin is failing, but propizamide's not. And if you're really interested in perhaps why this might be happening, you can talk to me at morning tea. The newer products that got in the mix now, Luxamax and Overwatch, we're just not picking up resistance in our survey. That doesn't mean it's not there. We actually know we've got resistance to both of those herbicides out in ryegrass populations. It's just that they're still very rare. So what the survey, I think, is telling us about ryegrass is it's telling us why we are where we are, but it's also telling us it actually doesn't matter where you farm in Australia. You're more or less in the same place, other than resistance to triflurin. And that's probably the one difference where geography seems to be making a bit of a difference. You know, despite lots and lots of triflurin use in Western Australia, only 4% of their populations were actually resistant to it. So for West Australian farmers, that's actually a bit of good news. They can still use that product. So that's ryegrass. For wild oats, the data I've got is around the Group 1 chemistry, where looked at Topic and Axial. And wild oats, well, you might have noticed in ryegrass there were lots and lots of samples, so 1,300 samples. It's the most common weed we came across. Wild oats is geographically a lot more limited in its distribution in terms of what we collect. So if we look here, New South Wales is dominating in terms of the number of populations, followed by Western Australia, and every other state is quite a bit smaller. We're also seeing a lot less resistance. Now, we've been spraying these herbicides on wild oats for just as long as we've been spraying them on ryegrass, but we're not picking up the same level of resistance. And, you know, Group 1 chemistry is a problem for farmers who have it as an issue, but for other farmers it's not really that concerning. We're also seeing a lot less resistance. Now, we've been spraying these herbicides on wild oats for just as long as we've been spraying them on ryegrass, but we're not picking up the same level of resistance. And, you know, Group 1 chemistry is a problem for farmers who have it as an issue, but for other farmers it's not really that concerning. Higher frequencies of resistance tend to be occurring in the states that had more wild oats. Okay, so Queensland, New South Wales, even though we didn't have so many fields surveyed in Queensland, so as a proportion, they had a high proportion of wild oats in their fields. They have more. South Australia, Tasmania, which have less wild oats, have less resistance. And there is differences between the Group 1 chemistry. So you don't actually lose all of your herbicides just because you get resistance to one herbicide. And I think this is where herbicide resistant testing can become a really important part of understanding what the problem that is occurring in that particular field is and how you might manage it going forward. Wild radish. The data in wild radish, again, we've got fewer samples. We're down to 136 samples now. This is dominated by what's happening in Western Australia. Okay, Western Australia had the bulk of the wild radish samples. And if we start with Western Australia, resistance in Western Australia to the Group 2 herbicides, to the sulfonite ureas, 86% of the population, so most of them. But Intervix, also a Group 2 herbicide, on wild radish is working to a greater extent. 2,4-D, half the populations in Western Australia are resistant. Diflufenacin, 20% of them are resistant. So Western Australia, a lot of wild radish samples, a lot of resistance. We move to some of the other states. Well, not much you can say about Queensland, Victoria and Tasmania because so few samples were collected, so don't get too excited about those numbers there. But generally speaking, there tends to be lower levels of resistance in New South Wales and in South Australia. But resistance is everywhere in wild radish. Okay? Every state has at least some resistance to one of these herbicides. So this is a weed where if it is a problem weed for you, resistance is in your future. If you don't have it as a problem weed at the moment, then you probably need to keep it out. So it's again, one of the themes that I'm going to have is this is about your population size. How bad your resistance gets depends a bit on how many weeds you have. The last of the individual weeds that I'll talk about is common sow thistle. Common sow thistle as a weed problem has come from virtually nowhere in Australia to be a fairly significant problem for a lot of growers. So it's one of those weeds that's actually changed its impact as well as getting resistance. So we had 500 samples because lots of paddocks have sow thistle in them. Almost everything had resistance to chlorsulfuron, particularly bad in sort of New South Wales, Victoria and South Australia, less so in Tasmania. A little bit of resistance to 2,4-D. Okay, 2,4-D resistance in sow thistle is now common enough for us to pick it up in our surveys. 5 years or 6 years ago, we'd have never been able to do that. So that's on the rise. And that's probably the big concern out of this data set is that phenoxy resistance in sow thistle is going to be the next really big issue. A little bit of glyphosate resistance. All of that's in New South Wales. So, this is a weed where we're starting to get some fairly significant resistance problems to some of the chemistry, some of the good chemistry we have against it. In the summer weed space, while we're testing a number of herbicides, the data set that I have at the moment is really just covering off on glyphosate, and it's not quite complete, but it's getting close. So these are the four key summer weed species that have been tested for glyphosate resistance. So flaxleaf fleabane, feathertop rhodes grass, ornless barnyard grass, sweet summer grass. And for all of those, glyphosate resistance is present. And for many of those, more than half the populations collected have glyphosate resistance. So this is a situation where, for some of these weeds, glyphosate resistance has moved very, very fast. So it's gone from being sort of a little problem for a few growers to now being across the landscape. For other weeds, it hasn't moved nearly as fast. So ornless barnyard grass, for example, is moving a lot more slowly than it is for those other weeds. And in fact, of those four weeds, ornless barnyard grass was the one where we first saw glyphosate resistance. So that's a really slow mover. So what we'll find is that some weed species will move quickly in terms of getting resistance and others will move more slowly. Understanding that helps us understand what do we need to do about this. When's the time that we really need to start taking action and saying we need to have a new management strategy for that weed? Those ones that are really starting to kick up, the time is now. Because by the time we develop that strategy, we're going to have lost that herbicide. And I think we can see that with things like feathertop rhodes grass. You know, we don't really have a fabulous management strategy for feathertop rhodes grass yet, but 97% of it's now resistant to our best herbicide. So what are we going to do? All right, so I want to finish off, and I'm going to talk briefly about, as I mentioned, about glyphosate resistance and phenoxy resistance and some of the recent understanding that we've got about that, and what they might tell us about how we should be thinking going forward and managing these weeds. So with glyphosate resistance, we've got now 21 weed species, of which half are summer weeds and half are winter weeds. And I know that doesn't add up, but South Thistle is both a summer weed and a winter weed, and it falls in both. Half of them are grasses and half of them are broadleaf weeds. So it's not like glyphosate resistance is really picking on any particular group of weeds and getting resistance in that weeds, okay? It's spread across all weeds and all agricultural sectors. Where we have our biggest problem are where we're getting those high numbers of glyphosate-resistant weeds in the system. And so that's things like, you know, fleabane, feathertop rhodes grass, annual ryegrass is really starting to get up there now as well. So they're the ones that are really causing us problems and where we need to have these alternative management strategies. One of the really interesting things that we're finding about glyphosate resistance is that for certain weed species, sometimes there's just one way they're getting resistance. So we've looked at, with James Herrwood from the University of Queensland, we've looked at hundreds and hundreds and hundreds and hundreds of feathertop rhodes grass, and they all the resistant individuals have target site mutations. And so that graph at the bottom there shows you, glyphosate rate for a number of different, these are feathertop rhodes grass populations, and what the response is. And what that target site resistance generally does is give you low-level resistance. So what's happening with these particular weed species, fleabane and feathertop rhodes grass, it's actually have innate tolerance to glyphosate at the moment. So you're putting on a relatively high rate to control them. So you're just getting control. Target site resistance is all you need to not get control. And I think the way you then think about, well, how do you manage that? Well, if it's just target site resistance to glyphosate, that means that you can do a whole range of different things without actually shifting that around too much. So glyphosate mixtures are an option. Double knot, really good option. So you can continue to use glyphosate on those weeds, even though they've got resistance, so long as you've got something else that's going to back that up. So that's that group. Then we have another group of weeds represented by these three grass weed species here, so barley grass, brome grass and windmill grass, where we don't get target site resistance. Okay, we've looked at quite a number of populations of these. We've never seen target site resistance. Instead, we get a thing called gene amplification. And what happens here is that these have multiple copies of the target gene. So they produce a lot of enzyme, and when you spray the glyphosate on them, it binds to all of these enzymes, gets tied up, but there's still got enough enzyme to live, and so they survive. And what we tend to see there is a bigger shift in resistance. And so the figure on the bottom there is showing the same sort of dose response curve, but this is for a number of populations of windmill grass now that have different numbers of copies. So this then opens the question, well, does that actually change how you might think about management? And the answer to that is, in fact, it does. And it does because of a really interesting phenomenon. So one of the things that we did with the barley grass was to start asking a question about what happens if you select this with glyphosate. So what we did is we took plants, we cut them in half, sprayed one half with glyphosate, didn't spray the other half. And the green ones with the dashed lines there were all the ones we didn't spray, and the blue ones were the ones we sprayed. This is the progeny, this is the seed off those plants. So it's exactly the same plant. All we've done is sprayed one of them with glyphosate. And we see that we get extra copies. So when we think about selection, we normally think about we're selecting individuals within a population. Here, we're actually also selecting on cells within a plant. When we measure this gene amplification, we measure the average for cells. These copies are not on the chromosomes. They're on little extra chromosomal pieces floating around. What that means is that if we do something like a double knock on these, we're going to just shift them to higher and higher glyphosate resistance. So here we might want to rethink what our strategy is. For 2,4-D resistance, so phenoxy resistance, we currently have 5 weed species, Indian hedge mustard, wild radish, common sow thistle, cape weed, and slender thistle. And one of the things that we've found in those species is we've found there are actually target site mutations in them. Where we get high levels of resistance to these to 2,4-D, what we find is it's a very particular target site mutation. It's actually a deletion within an AUX-IAA gene that then binds the herbicide and stops it having its effect. So the herbicide remains bound to the auxin binding protein, which is not affected, but you don't get the flow-on effects. These plants, you spray them with 2,4-D and they're hardly affected at all. So there are actually multiple AUX-IAA genes in plants. Okay, this is a big gene family. So this raises the possibility that whilst we might have 2,4-D resistance, we might be able to use other members of this family to bring in to help manage this. Because if they're binding to other AUX-IAA genes, this mutation is not going to affect them. And so it's probably a space to think about. What do we have that we might be able to bring in as mixtures or to move forward with other types of herbicide strategies where we might still get some level of control? Okay, so I've run out of time and I'd like to acknowledge all the people who contributed to some of the work that I talked about, and it's a really big list, including a large number of people from my team at the University of Adelaide. And I'd like to acknowledge the people who provided funding. GRDC provided the most to the work that I've talked about, but there's also been funding from Corteva and New Farm and from several foreign governments who've supported postgraduate students in my lab. So thanks very much.